Ah, yes, the nucleus in its natural habitat. As we can see, it is performing its normal functions, which will lead to the synthesis of proteins that we use in our body. Let's take a closer look. The nucleus contains the DNA for the cell. DNA is the genetic code for the cell and determines details about the cell. The DNA is condensed into chromosomes, which are then divided into genes. These genes are groupings of DNA that can either be turned on or off. The function of the cell depends on whether certain genes are turned on or off. Now let's take a look at the structure of DNA. Shown here is a close-up view of what a bit of DNA looks like. A single nucleotide is made up of three components: the sugar, the base, and the phosphate. The most important part is the base, which can be one of four things in DNA: adenine, also known as A, cytosine, also known as C, guanine, also known as G, and thymine, also known as T, along with additional nucleotide uracil, which replaces thymine and RNA. The bases pair across from each other, with A binding to T or U, and C binding to G. These bases determine what protein is made later on. Now we will move on to the creation of pre-mRNA. In order to synthesize mRNA, the DNA must undergo a process called transcription in which pre-mRNA is synthesized according to the sequence of the DNA. To determine the sequence of the pre-mRNA, RNA polymerase binds to the DNA and strings together nucleotides that are opposite of what is coded in the DNA. This is where uracil comes in, because instead of thymine opposing an adenine in the DNA, uracil is used instead. This process is called elongation after the elongating pre-mRNA strand. Once this process is complete, the pre-mRNA moves on to be modified before it exits the nucleus. Before the pre-mRNA can leave the cell, it must be turned into mRNA. This process is called modification. There are many modifications made to the mRNA during this process, one of them being a cap on the 5' end and a poly A tail on the 3' end. These components serve a purpose in binding to a ribosome later during protein synthesis. Another modification that has been made to the mRNA is the addition of introns and exons. The introns are really just junk DNA that are spliced out by spliceosomes during RNA splicing. The exons, however, stay on the mRNA and are used to express the genes and code for which protein to produce. After the complete modification, the mRNA is ready to exit the nucleus. You may be wondering, why is this mRNA so important? Well, mRNA is short for messenger RNA, which is what codes for which protein the cell needs to produce. Why it is called messenger is because it carries the message for the ribosome. Once the mRNA is out of the nucleus, it will make use of the 5' cap to bind to a small ribosomal subunit. This unit will move down the mRNA in the 5 to 3' direction until it encounters the start codon AUG. Next, the large ribosomal subunit will bind to the mRNA and the small ribosomal subunit. Both ribosome subunits are made up of rRNA or ribosomal RNA. This will cause a tRNA with the amino acid methionine to bind to the mRNA and start the amino acid chain. The start of this is called initiation. As the ribosome moves along the mRNA, it will read codons, which are groupings of bases that code for a specific amino acid. Upon detection, a tRNA will move into the A site, also known as the attachment site. The tRNA that moves in has an anticodon, which are bases that oppose the bases in the mRNA. This helps the tRNA bind and code for the correct amino acid. This is called translation, where the codons on the mRNA are translated into amino acids. The first tRNA amino acid will form a peptide bond with the new one and move into the P site. The enzyme that catalyzes the peptide bond is called peptidyl transferase and is located in the large subunit. This makes sense why tRNA is also called transfer RNA, because it transfers an amino acid to the polypeptide chain. Then, another tRNA will move into the ribosome and the previous two RNA will move over. The first one entering the E site will then move out of the ribosome to be reused. After the tRNA has been used, it is sent to aminoacyl tRNA synthase, where it is reattached to an amino acid. These amino acids come from the food that you eat and digest on a daily basis. The protein synthesis continues until the ribosome reads a stop codon, triggering termination that stops protein synthesis and completes the polypeptide chain. So, here it is, the entire protein synthesis process to produce this protein that will either be sent out of the cell or kept inside for intracellular use. Thanks for listening.